Today we're going to discuss the eukaryotic cell cycle and why it's so important to cellular life. You experience a huge number of cellular divisions in your lifetime. It's estimated to be around 10,000 trillion cellular divisions. Some of your cells are dividing almost constantly, others rarely or never divide. In life, we often go through phases that are defined by the focus of our interests and activities. For many years, we may go to school. For a few years, we would focus on our career. Then our personal life may take center stage and we spend time raising a family. Similarly, most eukaryotic cells go through their lives in one specific phase after another, a series of events called the cell cycle. Cells are born from other cells. They spend time doing the work of the cell, be it a blood cell, a nerve cell, a muscle cell, or any of the thousands of types of different cells. At some point, the cell may wish to divide. Before it can do this, it needs to copy its genome in the process of DNA replication, which we discussed last time. Finally, the cell may divide, creating two new daughter cells which would then become the parent cell in the next cell cycle. The cell cycle describes the production of somatic cells, which are the cells that make up the body. A different type of cellular division process produces reproductive cells. We call those cells gametes. There are two main phases in the cell cycle. Interphase, during which the cell grows and prepares to divide, and the mitotic phase, during which the nucleus and genetic material within the cell divides, and then the rest of the cellular content divides. Interphase is further divided into three distinct subphases. Gap 1. This is a period in which the cell grows and performs all its cellular functions, such as making proteins, getting rid of waste, etc. The DNA synthesis phase of interphase, the cell begins to prepare for cellular division by creating exact duplicates of its chromosomes via the process of replication. Before DNA synthesis, remember each chromosome is a long linear thread of genetic material. This thread contains a double-stranded DNA molecule. After DNA synthesis, each chromosome has become a pair of long identical threads, with each one containing an identical long double-stranded DNA molecule. Gap 2 is a phase in which the cell continues to grow and prepare for cellular division. This phase differs from Gap 1 because the genetic material has already been duplicated, and it's usually much shorter than Gap 1. The mitotic phase is the shortest in the cell cycle. The mitotic phase begins with mitosis, a process in which the nucleus of the parent cell divides. Mitosis is generally followed by cytokinesis, during which the cytoplasm, the organelles, and the rest of the materials in the cell are divided into two new daughter cells. Each daughter cell has a complete set of the parent cell's DNA and other cellular structures. It now becomes the parent cell in the next cell cycle. To give you a better idea of what this process looks like in living cells, I'm going to show you a video of sea urchin cells moving through the cell cycle. Sea urchin development has been very well studied, and urchins develop similarly to human embryos. So urchins are used as a model organism to better understand the mechanisms of cell cycle. First, you will see the eggs being fertilized by sperm swimming around the eggs. Then you will see the fertilization envelope lift from the eggs. This is to prevent more than one sperm from fertilizing the eggs. The sperm and nucleus fuse and cellular division begins. This first division is called cleavage. This happens approximately 2 hours post-fertilization. The cells eventually form a hollow ball called a blastula. These cells eventually invaginate, as you can see at the bottom of the video here, and these cells are beginning to form the gut, beginning with the anus, and eventually the mouth will form from the top of the developing embryo. You can see that the cell cycle is moving rapidly in this embryo. Okay, so thinking about this process of the eukaryotic cell cycle, the transition from one phase of the cell cycle to the next is triggered by specific events. For instance, a cell moves from gap 1 to DNA synthesis only if environmental conditions outside the cell are favorable for cellular division. Likewise, a cell begins mitosis only when the genetic material has been successfully duplicated. Answer the following questions to prepare for lecture next time. Why do you think the cell so tightly controls the cell cycle? What would happen if the cell cycle were not controlled? Second, how do you think the cell controls this process? Make a guess based on your knowledge of the macromolecules that we have discussed in class before. We will answer these questions next lecture and discuss the specific steps of mitotic division.